We are Zuri, Rizky, and Jax from Group A of Class S201, and we will be talking more about our ISS project on the effects of different sugars on the respiration of yeast and its enzymes. Firstly, for our introduction, so why did we decide to research on this topic? We were curious as to how different factors affected the rate of respiration of yeast, and whilst we were completing the project, we found it very fun, and that motivated us. Maltase and invertase are enzymes present in yeast. Yeast has many uses. For example, yeast is used in the process of making bread. Bread is a common carbohydrate that is consumed by many people, which gives their body energy. The process of making bread is simple. Yeast uses oxygen to release energy from different sugars in the process of respiration. When different sugars are used in the process of making bread, depending on how much energy is released, the bread will rise to different heights. During respiration, yeast takes in oxygen and carbohydrates. It then converts them into carbon dioxide, water, and energy. For our procedure, we place 5 ml of water, 2 ml of yeast, and 2 ml of sugar into a test tube. We covered the test tube with a cork that had a tube connecting it to a syringe. We then took down the readings of the syringe every 5 minutes for 30 minutes. As the yeast produce carbon dioxide during respiration, the carbon dioxide will push the syringe up, and we can measure the change in carbon dioxide by taking the reading of the syringe. We repeated the experiment twice for 7 different sugars, and this is the bar graph for the final average reading after 30 minutes. Here we can see that the red jaggery sugar, a type of processed cane sugar, gave the highest final average reading of 25 ml after 30 minutes, while the setup with sucrose sugar gave the lowest final average reading of 0.5 ml after 30 minutes. Our research shows us that the red jaggery sugar is a disaccharide that is a simple sugar soluble in water. The yeast was able to break down the cane sugar the fastest because it is less complex. We found it odd that sucrose has such a low reading. Thus, we repeated this experiment, extending the time past 30 minutes to see if we got any results. And here is the line graph of the reading of the syringe up to the 60-minute mark. Here we notice that after 30 minutes, the reading starts to rise very quickly. And while our research shows us that table sugar, sucrose, is also a disaccharide, it is made up of about 50% glucose and 50% fructose components. Thus, yeast needs to use invertase to break down sucrose down into glucose and fructose before it can get its food through the cell wall. Thus, we can conclude that while different sugars allow for different rates of respiration in yeast, red jaggery sugar allows for the fastest rate of respiration, and sucrose allows for the slowest rate of respiration out of our 7 sugars. However, we can also conclude that sucrose allows for a delayed reaction in the respiration of yeast. While yeast has many practical applications, such as in the food industry, or how fermentation of yeast produces ethanol, a biofuel, different types of sugars wasn't the only factor we planned to investigate. Another factor we had originally planned to investigate was how different temperatures affected the respiration of yeast. However, due to certain time constrictions, we were unable to do so. Finally, for our reflection, I feel that the biggest lesson we learned was learning from our past mistakes. We made many mistakes, especially during our experimental phase. However, we analyzed and reflected on those mistakes and made sure we didn't commit them again during further stages of the project. Thank you for listening.